Colorectal cancer stands as the third most prevalent diagnosis and the second most lethal malignancy. Notably, it's one of the cancers that can be effectively treated with the potential of cure, especially when detected at an early stage. Consequently, the importance of screening for colon cancer cannot be overstated. Presently, the commonly utilized screening methods include the search for blood in the stool, known as fecal occult blood testing, or the use of endoscopic procedure involving the insertion of a camera into the bowels to detect cancers. While these tests are valuable, it's essential to acknowledge their limitations and associated costs. Now, envision a scenario where we could harness the abilities of bacteria to navigate the stool samples and pinpoint specific cancer-related genes. What if this bacteria could not only detect the presence of these genes, but also identify the precise mutations within the cancer itself?
Good. So hello everyone, thank you so much for tuning in to another episode. My name is Roupen, I'm your host, Delta, and where we interview researchers, startup founders, and policymakers who are responsible of making a delta or a difference in healthcare system. Today, I have an amazing guest. I'm so excited from Australia, Dr. Worthley. Dr. Worthley earned a Bachelor's of Medicine, Bachelor's of Science with honors from the University of Adelaide in 2000. He furthered his qualification by becoming a fellow at the Royal Australian College of Physicians in 2006. His academic journey continued as he completed a PhD in 2010 and earned a Master's of Public Health in 2011, both from the University of Queensland. He expanded his horizon with a 4-year postdoc research at Columbia University, New York, where he worked with laboratories of distinguished figures like Professor Tim Wang and Professor Siddhartha Mukherjee. Returned to Australia in 2014, he established a new GI cancer biology laboratory at the South Australian Health and Medical Research Institute, starting a human colorectal organoid banking service, a new transitional mouse model of primary and metastatic colorectal cancer, and a synthetic biology pipeline of engineered bacteria to detect and treat colorectal cancer. He continued working as a consultant gastroenterologist in multiple public hospitals and private facilities in Australia. Dr. Worthley is an active clinician scientist with particular focus on inherited gastrointestinal cancers like GAPS, FAP, MAP, Lynch syndrome, colonoscopy quality, and colorectal cancer prevention. In 2019, he moved to Brisbane to become a partner at the colonoscopy clinic. Here, he collaborated with a team of highly skilled individuals dedicated to delivering a future where no Australian dies of colorectal cancer. I'm so delighted to have you, Dr. Worthley.
Roupen, thank you very much for reaching out to us. It's a delight to speak with you.
You recently published a paper in Science, and when I read the paper, my mind was blown. And that's why I wanted to interview you to explain this technology for everyone who's listening to the podcast in simple terms, and we can dive deep into the details of it. So let's start. What is bacterial engineering? If I'm just starting reading the paper or listening to the podcast, what does that mean?
Well, so engineering, if you dive into the words, engineering is about making something, an instrument, an engine, a machine, in order to solve a particular problem. And it's engineering, isn't it? It's not engineology. It's an iterative process. It's doing something. It's, fundamentally you design, you build, you test, you learn, and then you repeat. And that's the concept of engineering. So then we have bacteria. So the machine, the engine that we're creating is housed within a chassis, and that chassis is a bacteria. Now, how do you make a bacteria try and solve a problem? So how do you engineer using a bacteria to do that, or any cell? And all of the information, the code within a cell, the code that makes a cell, a specific type of cell, do a certain thing, become an eyelash, be able to fight cancer, is basically housed within its genetic code, the DNA that exists within the bacteria, and which of those genes it turns on at certain times. So the fundamental instrument, the the keys that we can manipulate in order to engineer a bacteria are the fundamental genetic code. And molecular biology is advanced to the point where actually manipulating the genetic code of an organism is fairly simple now. And so we have the ability to code a cell. And then we need to define, of course, what the problem is. And then the sort of the 4 steps are, how are we going to define the signal, the thing that activates the cell? How is that cell then going to process that signal via basically very analogous to electrical circuits, but genetic circuits that interact and work together? What's the best bacteria to contain this? What's the best chassis? And then finally, when the signal is processed within the right place via the right pathway, what is the output? So it's sort of signal, chassis bacteria, process, and then output. And people have been for decades engineering cells to address different problems. We used it as a way of detecting cancer.
Wow, amazing. So if I'm coming from an IT background, it's just like, it sounds like the way it's similar to engineering a software or a computer to do a certain task.
Brilliant. So, and it's written such in code like that. So it's a beautiful thing. Like I can't quite remember the story of the origin of biochemistry, but I think it was at MIT, and I think there was a biologist and there was a chemist, and I should know all these people's names, but they met in a corridor and then they just invented biochemistry. And the same sort of thing has happened with synthetic biology and engineering bacteria. It's been physicists and mathematicians and computer programmers and biologists who have all come together at the fringes, at the fingertips of these specialties to create something new. And it's, do you know what? I've been one of these people that is really at my fingertips for a lot of synthetic biology, but it makes it a hell of a lot of fun as well.
Wow, amazing. So I started reading your paper and terms started coming up. And I think one of the terms I asked myself, and if you can explain to the audience, is like, what is natural competence, horizontal gene transfer? What does it mean, and how it can affect the bacteria in the human gut?
That's a really cool question. It's a fundamental part of the paper. You know, if you were to say what is the fundamental part or the utility of the paper or the thing that we use, it is horizontal gene transfer. Do you know what? You know, genes can be passed. Genes are the fundamental unit of heredity. They're what give you your mother's laugh and your father's eyebrows, right? So, and they can be passed a number of ways. They can be inherited vertically from parents to offspring, or in the bacterial world, when one cell undergoes binary division into 2 cells and it passes from parent to daughter cell their genetic code. And that's called vertical transmission. There is also horizontal transmission. So this occurs outside of vertical parent to offspring inheritance. So bacteria, prudish as they are, they don't have sexual reproduction. So there is some value in gene swapping, transfer of different genetic information between cells. And the process that they do it is through horizontal gene transfer. So passage of one unrelated cell to another cell. And there are a couple of different mechanisms by which that can occur. There's conjugation, there is transduction, and there is also one called natural competence. Natural competence, the value of natural competence, the type of horizontal gene transfer, the natural competence is actually a cell that is able to look around its immediate environment and hoover up, vacuum up, suck up parts of cell-free DNA. So this is perhaps there's a nearby cell that has died or has been injured, and it's released some of its DNA into the immediate environment. And the bacterial cell a bacterial cell that has natural competence, this capacity to take up, absorb external cell-free DNA, is able to do this. And it often does it, in honesty, not only for gene swapping, but for nutrition or for nourishment, for other reasons other than gene swapping. However, it can take up, and particularly when there are genes or DNA that are similar to its own genome, it can actually integrate and use that external DNA into its own bacterial genome. There are, obvious, and some of your clinicians will know, in terms of antibiotic resistance, this can be a mechanism by which antibiotic resistance is conveyed. And in fact, the inception of this project followed a presentation that was just about that. So I was at UC San Diego, Jeff Hasty's lab meeting, and Rob Cooper, who's the first author the equal first author on the paper, was presenting work on antibiotic resistance, and he had Acinetobacter bailey, A. bailey, which is the sort of the star of the show, which is a very well studied naturally competent cell. So this bacteria is a real maestro at sucking up external cell-free DNA. And he showed this really cool video. The Hasty Lab always does very beautiful self-evident science where you can really understand the methods and the results within a few frames of a video. And he basically had one green cell and one red cell. And the green cells were resistant but were getting eaten and passed on directly the antibiotic resistance gene to the red population, which then exploded in a bloom of red and green becoming yellow. So it was this sort of bacterial natural competence, antibiotic resistance mechanism that he was presenting. And looking at two cells, and you would do this in your own life, whether clinically or in the lab, you just, there's the obvious analogy from your own life, whatever it is, and you can just apply, look, one of those cells could be cancer. So how could we make that work? And that was the sort of the discussion at the end of that lab meeting. So horizontal gene transfer is the passing of genetic material from one cell to an unrelated cell fundamentally. And this happens it's very commonplace in the microbial world.
Wow, amazing. So like for us as human beings, we can only pass the genes to our kids and children, but bacteria can pass it to each other, can take it from the cells around them, even if they are human cells in simple terms.
Absolutely, or from viruses, transduction from bacteriophages that infect them and carry other genetic material. So, yes, there is. Although there is not sexual reproduction, there is still a hotbed of genetic swapping. Lots of little keys in bowls all around the bacterial world.
All right. So I think we talked about the first two definitions, and now the third definition will help us understand the paper better. Can you tell me a bit about like what's CRISPR and what's Catch, the technology based on CRISPR, in simple terms, and then we can jump into the steps of this paper to make it more sense.
That's the critical your third part to it, and you've done that perfectly. So CRISPR is an ancient means of adaptive immunity, and of course Jennifer Doudna and Emmanuelle Charpentier won the Nobel Prize for discovering this entity and then being able to utilize it. Fundamentally, it's a way of a bacteria being able to recognize and then attack viral particles that they've been exposed to in the past. So CRISPR stands for clustered regularly interspaced short palindromic repeats is what CRISPR. So it's these repetitive sequences that were found within the bacterial genome. How that can then be used, it liberated the capacity for genetic engineering in obviously in a unique and immensely powerful way. And like polymerase chain reaction was impossible to remove from a molecular biology laboratory from the very first moments that I was ever a scientist. CRISPR is now just inherently fundamental to all aspects of molecular biology over such a short time frame. So the way that we utilized it is that, so CRISPR was, I think, first described in strep pyogenes within a specific bacteria. However, lots of bacteria have a similar system. And Acinetobacter bailii is no different. So it has its own CRISPR system, which Rob Cooper and Jeff Hasty had been working on prior to this work. What that provides is, therefore, one is able to design a specific code that then is flanked by something called a protospacer adjacent motif, or a PAM sequence. PAM sequence is like two-factor authorization for the enzyme to cut a certain site. So you have the sequence, but you also need to have this other additional three-letter sequence. Acinetobacter bailii is NGG. So you have this third little sort of cuff of code. And so what that allowed us to do, so that's the fundamental mechanisms of CRISPR. A specific genetic code by a guide RNA can attract a DNA's cutting enzyme. Why that's important for us is that we wanted to define a cancer-specific gene. The tricky thing about cancer-specific genes is they're often very similar to normal genes. So all of a sudden, you need to define something sometimes on a single base pair change. That is the case for one of the most important oncogenes in human cancer called KRAS. So KRAS G12D means that at a specific place, codon 12, there's a change in the amino acid residue from glycine to aspartate, G12D. But the actual genetic event that generates that change in amino acid is a substitution of guanine to adenine. So G to A substitution. One single base pair is the difference between normal KRAS gene present in every single cell of your body versus cancer-causing KRAS. So that seems like a hard thing for a bacteria to do. However, CRISPR is able to provide single base pair resolution if one can design a guide where that second factor authorization PAM site overlays that mutation site, which is how we designed this approach. So I'm getting a little bit ahead of myself, but let's, if there is a KRAS gene that comes in and is integrated into the bacterial genome via a mechanism that we may allude to later, if it's wild type, then the PAM sequence is maintained, the Cas-associated protein comes in, cuts the DNA and kills the cell. If, however, the DNA that comes in is mutant KRAS, it then has a single base pair substitution that all of a sudden destroys that PAM site. All of a sudden, the code that is required for the guide RNA to bring in a DNA cutting protein is lost. That bacteria that's taken up mutant DNA is then preserved. And if it has a means of selection, it will proliferate. But only those that have taken up mutant DNA will be able to survive. And so that was a very lucky, like essential, but also it was very lucky that Rob had been working so closely and so hard on Acinetobacter bailii CRISPR system. We were discussing this, this problem, when we were already down. It was obviously something that was coming up, but we thought that the first bits probably wouldn't work. So we presumed that this wouldn't become a major issue. And Rob was like, well, actually, I've worked on that before. It's just a single base pair mutation. I'm going to be able to engineer it, provided it lies within a PAM that will be able to cut the wild type DNA. And the PAM sites are actually very regular. So that actually it was a very high likelihood that KRAS G12D, or in fact any codon 12 mutations would have a PAM that would overlie it. So that was a confusing, rambling answer, but I hope that paints some of the CRISPR value.
So if we're going to say in simple words, CRISPR, it's like a scissor that cuts the DNA in a specific area and help us to transfer that DNA to the bacteria. Would that make sense?
It's perfect. All of a sudden, it is the molecular scissors. Absolutely. Wild type KRAS comes in, it's paper. Mutant KRAS comes in, it's a rock. So if you do that with rock, paper, scissors, that's the sort of the analogy.
Now that makes sense. And now we have the bacteria who wants the DNA, and now we can tell the bacteria which DNA to take and integrate into its own genome.
Precisely.
And I'm not sure if we touched base about the technology that you also mentioned, the catch, and how it's related to CRISPR, and is it the same thing or not?
Yes, so catch is actually was a catch-all for the entire technology that we described. So, a cellular approach to characterize horizontal gene transfer. So this was essentially the concept of using a competent bacterial chassis. It was the concept of then using bait DNA within the bacteria. So natural competence can occur, but the likelihood that the DNA that is taken up would integrate into the bacterial genome is based largely on how similar the bacterial genome is to the target DNA. That's largely so that, bacteria can sample genes from similar bacteria rather than, totally different bacteria that may not have genes that are going to be of value to it. But so what we did in that engineering is, so naturally competent A. bailey takes it up. That's just A. bailey being A. bailey. What we did is we engineered A. bailey to have a very long segments, flanking segments, that were very similar to KRAS, human KRAS gene. So all of a sudden, if it took up KRAS, then it was able to more readily integrate. That was the first sort of invention, not an invention, but application, which was really using homologous recombination. So DNA that looked like the target DNA. And then that would be able to integrate. There were some both proof of concept and then more application-driven ways that we were able to generate the readout. So initially we did it such that the target DNA that was taken up actually corrected a defective antibiotic resistance gene that lay between these flanking regions. And then subsequently we did a different approach whereby natural KRAS DNA, when it was taken up and integrated, it removed a repressor to a subsequent antibiotic resistance gene. And that was the final sort of step whereby a totally natural bit of DNA within a target tumour was able to activate our bacteria. It's important to know for the scientists and the clinicians will rightly want to know is that some of the experiments, particularly at the start of the paper, are really proof of concept experiments where we had engineered both the biosensor, the bacteria, but we had also engineered the DNA within the tumour. That sounds sort of a bit counterproductive, and of course there was a whole lot of discussion about it. But the reason we did that is we really wanted to test the concept that horizontal gene transfer could work at all in a complicated system. And we felt that the likelihood of it working was very, very low. And so we designed a system as a proof of concept to ask that specific question, understanding that we would need to do further engineering in order to apply it in the clinic.
Amazing. So far we defined three important terms, and then so we talked about how bacteria can be engineered and we can change the DNA, and that's bioengineering. And then we said how bacteria can take DNA from its own peripheral tissues or cells. And then we talked about how CRISPR, how we can cut the DNA in a specific area and in a specific mutations, and can make this bacteria take this DNA and integrate it into its own genome. Now, my question is, if we can, in simple terms, summarize the entire experiment after we build the building blocks quickly, like what was the experiment, what did you do in it, and what were the results?
Lovely. So the way that you're able to get people into slightly grandiose experiments is to go stepwise. You've got all of these go, no go signals along the way that you can abort and save face before you go into very, very large and costly laboratory and animal house and other experiments. And so it's like sort of breaking bad news to someone. You do it sort of slowly and
exactly slowly.
Yeah. So the first question was, so could this work at all? So the very first experiment we did, which was to engineer the bacteria, the biosensor, to have all of its parts so it could do its job. But the target DNA was initially only the DNA. It wasn't the needle in the haystack. It was 1 trillion needles is what it was. And so it was what's called plasmid DNA. So it's largely just the purest essence, the Chanel No. 5 in a small glass vial of target DNA that we marinated the biosensor in. And so that's a very easy, very straightforward in vitro experiment. And then that worked. So when you just had the target DNA and you mixed it with the biosensor could detect the target, not tumor DNA, but the cancerous DNA, the mutant DNA. The next was then, okay, well, if you take then cancer cells, no other cells, no stroma, no microbial DNA from the luminal soup, just cancer cells grown in a dish, and then you lyse them, then you purify their genomic DNA. So now you've got the target DNA, but all the other genes. That also worked. And then we took cells that we sort of beat over the head, what's called cellular lysate. They've got all their other proteins and other things, and marinated that, and that worked. Then we just co-cultured cells together, cancer and bacterial cells, and then that worked. Then we used organoids. So organoids, you know what, they are also very standard in a typical cancer biology lab, a slightly more complicated form of cellular culture, more representative of in vivo tumours. And once again, we did that with co-culture, and then that worked. Then we mixed the biosensor with some mouse poo and spiked that with target DNA. And that was a very interesting experiment that, now with so much competing, not even non-target DNA, but just sort of microbial DNA from all the bacteria in the mouse lumen. And then that worked. And then that was the go, no go signal to then consider doing this in a mouse model. So mouse models from all of those, that they are increasing increments of complexity, but then to go from sort of spiked mouse poo to administer something to a mouse, that's a dramatic increase in complexity. It's an essential increase in complexity. So, but people bring threads from their previous lives. So Rob had obviously brought horizontal gene transfer Jeff has, one of the world's greatest synthetic biology labs, and he's brought all of this to it. What our lab brought to it was that we had, one of the models that we generated when I moved back to Australia was a modular model of colorectal cancer in a mouse. So we have organoids harvested from a mouse, genetically engineered in certain ways depending on the tumour we wish to model. And then via mouse colonoscopy, you can inject that into the mouse lumen.
Wow.
And then sort of over 6 to 8 weeks, it develops a primary tumour. There are lots of different mouse models of colorectal cancer. The reason why that one was good is because the tumour itself is modular. So it's not a genetically engineered mouse model whereby the mouse tumours develop via an unalterable genetic sequence. We're actually able to engineer, again, that word, the tumours to express the genes that we want them to. And that was important for this
particular. Able to go with the colonoscopy into the mouse and inject some cells that can make the tumor that you want with the specific mutation that you want.
Exactly. So that's, it's a cool, it's a nice, it produces lovely videos. It is, when you're presenting to a clinical audience, it's obviously attractive, but it's valuable, particularly. And when I came back, in the Columbia lab, we didn't want for resources in terms of funds and animal house budget and size. But when I came back, I didn't have a whole lot of money. And the reality is that it's a lot cheaper too. If you want to study a range of different, it has limitations, but if you want to study a range of different genetic types of tumour, to have something that you can engineer in vitro and then deliver into a generic mouse has value when you're wanting to study different pathways. So, for a range of reasons, largely because I was a bit skint and wasn't flush with funds, we chose that as being the model that we would develop. But then for this particular project, you had to have a modifiable genetic system in the proof of concept. You had to be able to design a tumour that housed important target DNA that we would then be able to study in our first mouse experiment. So we each brought those things in. We then delivered, and the biggest bang for your buck, although, and once again, it is a far cry from the clinic and how we would do it ultimately in humans, but we delivered it via an enema, the biosensor. How do you get the most bugs around a colorectal tumour that you've injected via colonoscopy? And that's to put a high number of them rectally into the mouse. And so that was, once again, it's a proof of concept, not how it would be applied. And there are developmental aspects that we would need to do for it to be an orally deliverable biosensor. Any biosensor, any engineered bacteria with a target within the gastrointestinal tract, in my opinion, needs to be delivered orally for it to be an effective clinical solution. So we are not at that stage, but that was the experiment. Mouse with tumour, mouse without, sort of a case and control type study, delivered biosensor, and then we collected that and then found that theoretically those biosensors that were exposed to tumour should become resistant to a certain antibiotic. We plated those and found just that. They turned green, they're antibiotic resistant, and only in the tumour, only the biosensors delivered to tumour-bearing mice. So, we were able to show that we were able to find all of the tumour-bearing mice, and the sensitivity and specificity was perfect in a very small, in a mouse model in our hands. So that was the stepwise, very simple, the simplest possible experiment through, not to the most complicated experiment, but to a complicated preclinical model to show that it could work, in the vagaries of stromal cells, other inflammatory cells, the microbial soup, So that was a lovely moment when that ultimately worked.
Wow, this is mind-blowing. This is like amazing. When I read the paper, I was very impressed. I'm still impressed. I'm just like, so like in simple terms, you were able to bring some mouse and some mice, and like you were able to make a tumor with a specific mutation grow. And then you brought bacteria, you told the bacteria, go and detect this specific mutation in the mouse model, and the bacteria was able to do that.
That's right. That's right. So
Wow.
the mouse model that we published in Science, it had modified target DNA as well as being the proof of concept demonstration that was able to correct antibiotic resistance within the biosensor. So it was tumor DNA that was released from the tumor into the lumen. That's where it met the biosensor, which was able to take it up, incorporate it, activate a circuit within the bacteria to have the readout. So yes, so it was an exciting sort of development.
Amazing. So now, after talking about the experiments, let's talk about the applicability.
Cool.
Currently, like, let's say, can you tell me a bit, and can you tell the audience a bit, like, how do we screen people for colon cancer, and what are the problems with the screening methods?
That's such a great question. And there's, you know what, there is a whole lot that is great about colorectal cancer screening, both in the US, Canada, and in Australia. And you know what, there is so much good at the moment that, you know, you don't want to dissuade anyone from getting involved. The key approaches are either that sort of basically for people that are at average risk, that don't have any symptoms, that don't have a high family history, or a high risk from family history. Basically, they come along at 45 years of age. The actual ages vary a little bit, but the Australian recommendations are now 45. I know the US ones depends on which society you're listening to, but 45 is broadly considered to be the time of starting screening for colorectal cancer. And then there are a couple of different approaches, and you know, several of the approaches are endorsed. Sometimes there's two-step screening. So that might be either looking for microscopic blood that's released from the tumor into the bowel lumen and then into the stool, into the poo. It's called immunochemical fecal occult blood testing. If it's positive, you then go on to colonoscopy, two-step. There's also stool DNA. Cologuard is a very popular test in the US that combines testing stool DNA. So this is abnormal DNA from the tumor, as well as immunochemical fecal occult blood testing. So they sort of combine those two, and we can talk about why that's important. And if that's positive, you go on to colonoscopy. Also, there is direct screening with colonoscopy. And then there are some more other peripheral sort of optical views, such as CT colonography, where if a polyp is detected on basically an X-ray, you then go on to colonoscopy to make a histological diagnosis. So you sort of go from screening on the basis of pre-test probability on the basis of age largely, and that needs to improve in the future, but currently that's it. And then you go to sort of somewhat increasing that pre-test probability further by blood in the poo, some abnormal DNA or blood in the poo, or an abnormality on an X-ray, to then further increase that pre-test probability to get to colonoscopy, or you go straight to colonoscopy. Now, the benefits and risks of those two, the benefits and disadvantages of those two, the first one is participation. Not necessarily are all of these things resolved by our application, but participation is the biggest issue. There's a very widely promoted New England Journal of Medicine article not that long ago, which really showed that some colorectal cancer screening, although reliably shown previously to improve cancer-specific mortality, had less than stellar advantages when you considered on an intention to treat basis. But when you drill down into those that did it, it's really valuable. So the problem here, like in America and like in Canada, is that a lot of people, particularly at the age of 45, aren't tuned in to cancer screening, one, and if they are, they're not super keen on testing their poo or having a colonoscopy. So, participation is important. Now, why is participation poor? Well, part of those factors are just inertia, feeling like you're too young. The grey hairs haven't come in quite so much as they have in me yet, so you think you're young. But then there are other real, non-psychological things like cost. That's a really important one and can be limiting depending on where you live. Certainly, inconvenience is a huge one. And although colonoscopy can be delivered very simply, conveniently, and and with determination, affordably, it's a day off work, and that part of it can be difficult for some people. And you know what? Colonoscopy is a very safe procedure, but there are theoretical risks in terms of complications of any invasive procedure requiring sedation. So
yeah, I completely agree. Like my mom also, like she was gonna have a colonoscopy, and just the preparation for it, like getting prepared, the drinking that fluid and having all the diarrhea 24, 48 hours before.
Yeah.
My girlfriend had the colonoscopy, and the fasting is sucked. It wasn't a happy experience at all. And then like someone drive you have to take time off work, so there's some financial toxicity as well. So there are tons of things that comes with colonoscopy.
We are an Amazon society. Like, we are a customer service society where, healthcare needs to be delivered so that you are delighted with the actual health interaction. And you know what, as much as we try, and I work at a place called Colonoscopy Clinic, we do a lot of hard thinking around this, but you know what, we are not there. We are not there at having a delightful experience. And people tell us because they don't get involved in participation. And so they don't participate in screening, and then they present when they develop symptoms. And
exactly.
The reason colorectal cancer is such a great one for screening is that early colorectal cancer, whenever I diagnose early colorectal cancer, particularly from screening, it is a story of success. These people go on. They may require surgery if it's cancerous at the time. They may, if they had just have an advanced polyp, it's curable at colonoscopy. But then almost all of them, more than 95% of people at stage 1, will live following surgical resection alone. However, as it advances and becomes more advanced, spreads to lymph nodes, then all of a sudden adjuvant chemotherapy is required.
That's when it becomes hard.
Spreads to liver, and then that's when the mortality is dramatically impaired. So you either bring diagnosis earlier to earlier stages or advanced pre-invasive disease, or you improve survival in advanced diseases. They're the way you stop people dying of colorectal cancer. So for all of those reasons, you know what, there are new things on the horizon, and it's important to talk about these other new ones. So wonderful companies like Grail and other screening services of the blood, which is far more convenient as a sort of an evolving new first step as a means of molecular risk profiling to see if they can find tumour DNA within the blood to then sort of initiate colonoscopy. So what are the benefits what are the benefits of having a cell do those jobs? Well, do you know what? So laboratories and in vitro testing is not too bad. Do you know what? It is not so dreadful. And Cologuard as a test, which combines stool DNA and fecal occult blood test, has pretty good data around it. The advantages of a cell-based system there are two advantages. I see the strongest ones. One is that it was able to detect DNA in an unprocessed sample. So what does that mean? Well, it means that you don't need to extract it. You don't need to send it to a lab. You don't need people in white coats to use reagents to process it. By using a cellular biosensor, all of the sophistication goes into the cell. So you miniaturize a bit like a pill cam. I'm a
Yes,
gastroenterologist.
yes.
It miniaturizes a movie, a cinema set within a pill in order to take a video. A cellular-based biosensor miniaturizes a laboratory, and so it's able to reduce the cost and complexity of processing a sample. And do you know what? That may not always be of dire importance in Detroit or in Brisbane or in other, but certainly as you get into more resource poor or more remote settings, that can be really valuable and has real potential.
I completely agree.
The other thing that's nice that a laboratory test can never do with a cell-based test, and it's part of why colonoscopy is a pretty good test. You can detect a precancerous polyp, and then you can remove it. You can find and respond because you've got an organism, a couple more cells than one, but you've got an organism in the middle of that process. Maybe there might be machine learning, artificial intelligent ways of cool machines doing that in the future. But at the moment, it requires an organism to detect and then respond. And CAR T cells are a perfect example where they detect and then they kill a cancer. And they can do that because they're a cell. And so we're gastroenterologists are cancer preventionists. So, where this was very fortunate to get published in Science because it's a cool new signal for all of
technique,
for synthetic biology, but maybe the clinical application where this would come into its own is going to be responding to that signal. We didn't publish that, and I don't want to suggest that we didn't show that in the paper at all, but that's the end point where you have detection of unprocessed sample, which we can do. DNA is a cool new signal, but then you can respond the cell through the circuitry. You can respond at the time and at the place of disease detection, and so that's years away, but that is the promise of that technology above those others.
I just want to add one point. Even like in resourceful areas, like here, like in Canada, like my parents are in Canada or in Ottawa, and like I'm doing my fellowship in Detroit. Like for example, in Canada, the government here, it's they send this fecal occult blood tests to my parents, and they have to do it and send it back. Like it is still a test that takes time, takes effort. So like you have to collect your poop in a specific way, and you have to put it in the testing kit, and then you have to send the testing kit back to the government or like the health companies who are running the tests, and they have to do the test, and then they will give you back an answer. Like even in like first world countries where everything is there, like we still have like there are lots of steps to do. For example, like I can't really count what I eat every day because like I need to manually input my food, which is like
stupid. That drives me nuts. Yeah,
right. So it's the same thing here. Like for example, my mom sometimes, like, which I don't recommend, like she skipped doing the fecal occult blood test because like it's lots of work for her, and that should not be the case. But now with this technology, and that's what got me excited, we can bring the entire, we don't need all that processing. We bring the bacteria, put the bacteria in the poop, and tell the bacteria, is there cancer DNA or no? Boom, we have the answer.
Yeah, so there is, it promises some cool avenues. Absolutely, I agree with you, and I'm delighted that you're delighted too.
Oh yes, I'm very excited. Like the future is here. We need to change how we practice medicine.
Yes.
Here. So that being said, now we're talking about specifically like colon cancer.
Yeah.
Do you think that with bioengineered bacteria, we can start detecting other types of cancers?
That's a great question, and obviously the answer is yes. So, it is, the DNA input is modular. It can be any DNA sequence that we are bringing into the biosensor, and because that's just regulated by the sequence of the homology arms within the biosensor. So at the moment for cancer, it is limited to, or at least we've only problem solved for hotspot target mutations. So in order to utilize this CRISPR technique, we couldn't detect, for instance, the deletion of a tumor suppressor gene. APC is a classic one. So our system wouldn't be able, so we could detect KRAS G12D or BRAF V600E, also present in melanoma. I mean, we could detect specific hotspot mutations, but some other genetic events in cancer, chromosomal instability, etc., we haven't got a solution to as yet. But absolutely. So, and I don't think it's limited to gastrointestinal cancers per se either. The way that it becomes a bit more powerful, like one mutation is cool when previously no mutations could be detected by bacteria. But the way that you make it a bit more powerful is to multiplex it. So, and Cologuard, it does look at KRAS G12D, but it looks at other mutations as well.
Yes, exactly.
And then so you can build up and overlap in order to increase your breadth as well as your specificity for certain tumors. Gastrointestinal tract is an obvious one because it's filthy with microbes in health. But with a complementary chassis species, you could foresee a time where the stomach, the esophagus, the nasopharynx, the genitourinary tract, the skin, etc., could be addressed in the same way. And the other thing to say is that yes, cancer is cool, important. Cancer is important. It's very uncool, but it's very important. But DNA is important in a whole lot of things. So the other classic clinical scenario would be an infection. So infection and cancer are clonal disorders. They start with a specific cell that proliferates and defined genetically. So, that's also some low-hanging fruit, particularly in the gastrointestinal tract, either to record or respond to certain infections, certain microbes, particularly those that may be fairly poorly harvested or cultured from a stool sample because they reside higher up in the gastrointestinal tract. So, it's cancer, it's infection, it's further development with multiplexing, it's proof of concept of being able to both detect and respond. They're the next clinical steps. There are also a couple of other sort of kooky ideas. The guys in the biology lab do a lot of environmental work where bacteria responds to heavy metals and other things within water sources, and certainly anywhere where DNA and life and measuring it is important, whether that's an algal bloom in a nuclear cooling pond, or there are other sort of non-medical applications that we don't necessarily have the immediate bandwidth to utilize, but we hope that we might be able to exploit in the future.
For our listeners who don't have like the medical background, like they don't understand the pathobiology or pathophysiology of colon cancer. So when colon cancer arises, usually we can detect some genetic mutations. And what we've been talking about in this podcast and this video is like we engineered bacteria to detect a specific type of mutation, but there are tons of other mutations that we are not able to detect currently with bioengineered bacteria, but we will hopefully be able to do that in the future. And these mutations, for example, like one of the same mutation that can cause a colon cancer, it can also be linked to other types of cancers. And that's why we test for these mutations. And there are some people, for example, I saw a patient who had the CHEK2 mutation a while ago, and patients with CHEK2 mutations are exposed, or they have higher risk of developing, for example, breast, thyroid, colon, and other types of cancer, or ovarian cancer. So now cancer, it's not the cancer, But the question is not detecting a cancer. The question is detecting the genetic mutation with bioengineered bacteria that can go and tell me, okay, so this person has this mutation and they have high risk of developing XYZ cancers, and that's where the future lies.
Absolutely. So yes, and to be able to link a genotype complementary response is an attractive sort of additional. You've got the laboratory and then the physician within the cell.
Got it. Okay, so the last question, and
Yes.
so when we think about this, like, do you think from your perspective, are there any ethical considerations or safety precautions that should be taken into account when you are using this technology, bioengineering the bacteria to detect the specific genetic mutations?
So the answer is absolutely yes. And so the however, what I would also add is that engineered bacteria, engineered cells are commonplace, not only in trials, but CAR Ts are a classic example of engineered cells that are FDA approved and being used today and are saving lives in young people with ALL. So there is absolutely, and I'll go through and list the key sort of regulatory requirements, or at least hurdles that we need to get through, and potential concerns. But I just wanted to preface by saying that engineered cells are here, and they are being used. So although, and a couple, people have talked about this as being sort of almost like scientific, science fiction. And that's the way I feel about a lot of cellular therapies. Like, these things that are now commonplace to us, whether it's stem cell transplantation, organ transplantation, CAR T cells, in vitro fertilization. How crazy is that? And how beautiful. So there are all of these, there are these incredibly commonplace is not the right thing, but there are these miraculous cellular therapies that we use already, that we take for granted. However, the new kids that come along absolutely need to be vetted, and the biocontainment for bacteria is, of course, always a huge, and increasingly in recent years, the containment of biologically modified organisms, is appropriately, very carefully scrutinised. What are those regulatory requirements? Well, you need to prove that the chassis species is safe within human trials. So Acinetobacter bailey is largely non-pathogenic in immunocompetent people, but we would need to look at how does it go in normal healthy folks, even without the engineering. So to be delivered in capsule form in the dose that we're going to be delivering it in. And then the genetically engineered form within a patient group, so a phase 1 trial in people with the known mutation, to see if we're able to detect and respond. We need to be cautious and measure and ensure that, just as there is DNA swapping in between tumour and bacteria, that there isn't swapping between bacteria and other bacteria. That makes the readout of antibiotic resistance a less attractive readout than other less concerning readouts. And there are other ways of metabolically selecting for cells that are of no antibiotic transfer risk. And then it needs to fundamentally be shown to be doing its job better than the current gold standards, and the gold standards are good. So there is a series of hurdles to get over. The safety ones are important. Biocontainment, antibiotic resistance, just the nature of the chassis species, how it affects one's native microbiome, however, companies like Synlogic and other companies are using engineered bacteria now for metabolic diseases such as PKU. And so, this is stuff that is happening. And what's lovely, it's great to be the first, but it's hardest to be the first. And I feel like we're coming in at a time in bacterial engineering where some of the heaviest lifting has already been done by some other great pioneers in terms of the regulation. So, it is still challenging stuff, but it has precedent, and that always helps when trying to deliver something of value.
Yeah, I completely agree. I just want to quote a great book I've been reading recently. I'm very interested in entrepreneurial books. There is a great book. It's called 0 to 1, and it's by Peter Thiel. And like one of the concepts, he says it's not about the person who gets first into the market. It's about the person who go to the market and find something that can last for 10 years. So I think this is a great time to introduce this and to be in the market as well.
That's a very cool. I'll read that book. That sounds like a cracker. That's a very good insight.
It's a really great book. It's a really great entrepreneurial book for sure.
Rupert, may I just make one further additional thing that I'd love for you to incorporate? So, this is synthetic biology, which is really, as I said before, it's at the fingertips of other specialties and other expertise. And I just wanted to acknowledge, I've mentioned some names already, but Professor Jeff Hastie, pioneer of synthetic biology, a giant in the field, which I'm very lucky to work with, and was really helped drive this project. His originally postdoc and now scientist working at UCSD, Rob Cooper, who was equal first author in the paper. I won't go through every single author, but I wanted to also mention Josephine Wright, who really did the lion's share of the actual experimental work in the lab, and also Suzanne Woods, a wonderful colleague and scientist who has been, who I started working with not long after I returned to Australia, and all of those people together, and then the additional authors, were all essential to getting a sort of a complicated, multifaceted study through.
Thank you so much for everyone who was part of this study. It's been amazing just to read about it and see it in the news and see how everyone's talking about it. And hopefully this will be the future for us, for humanity, because like no one should die from colon cancer. And hopefully with bioengineered bacteria, we can make this happen in the future.
Thank you so much for your time, Roupen. Have a lovely day.
Thank you so much.